and welcome to Track 3, New Approaches in the Genetics of Disease for LabRoots Genetics Virtual Conference, April 20th to 22nd, 2021. I'm Rasika Mathias, and I'm excited to talk to you today about sequencing the genome from genotype to phenotype and a few extras in between. It's 2021 and time to say happy 20th birthday to the human genome, 20 years since its sequencing. And it's perhaps important to take pause and just think about how far we've come. 20 years ago, we were looking at complex diseases genome-wide, but we were using panels of genetic markers on the order of about 300 markers. Today, we're looking at complex diseases genome-wide, but we're looking at millions and millions of SNPs, thinking back to this recent publication from NHLBI's Precision Medicine program, 400 million SNPs, 53,000 individuals. We've come a long way, and today, if you run a NovaSeq for 24 hours, you're getting 3 terabases of readout in a single 24-hour period. So that's pretty incredible to think about, and the cost of the human genome obviously has declined rapidly. Along with this, we are now generating extensive amounts of next-generation sequencing, and in the next 30 minutes or so, I'm going to sort of walk you through an example from NHLBI's Precision Medicine program, TOPMed, where we're leveraging these genomes to call variation corresponding to the dynamic genome, or telomere length and chip, if you will, and really hone in on the importance of a multi-ethnic representation in large precision medicine efforts. The GWAS catalog today has close to 5,000 publications, well over 200,000 trait associations. But really, it's important to think about where this representation stands in terms of human populations. And a large fraction of this research actually really is focused on populations of European ancestry. We've made some headway in bridging these gaps. Here's an illustration, 2009 to 2016. We've gone from 96% down to 81% European representation, but really the critical bridge here has been Asian populations. Populations of African ancestry and Hispanic Latino populations are very poorly represented still. What's interesting when you think about this is that, particularly for African ancestry populations, the little blue parts of these pie charts noted here, while we represent in these studies a small fraction, these studies actually do contribute a much larger proportion in terms of the discoveries made. Why does this matter? Well, let's think about the promise of the polygenic risk score and precision medicine opportunities. The polygenic risk score is our means to use and leverage genetic discoveries to risk classify individuals and sort of really think about intervention and medical interventions and therapies targeted for individuals at high risk that can be almost diagnosed before the onset. And that's this whole PRS framework. But what we do know is that there's really non-transferability of the PRS, especially as the distance between discovery and application populations grows. Even for something as simple as height, as illustrated in this really elegant example, we know we do really poorly when we take discoveries of genetics in European populations and propagate them onto non-European populations. So with respect to the lack of representation and multi-ethnic nature of these data sets, there are many issues we grapple with. For or here, disease biology remains not well understood. We're not sure if these discoveries in European populations are being replicated. We're potentially missing associations with disease traits because these are discoverable only in non-Europeans. And to be honest, the further away we are from the discovery population, the less likely we are to be able to predict risk accurately. And this is perhaps the critical piece here from a precision medicine initiative and all of these large amounts of biobank data and large amounts of next generation sequencing data that we're amassing. So I'm going to use this sort of story, which is, a story in the era of whole genome sequencing, and actually a bit of a lecture on the telomere chapter here with difficulties that we mastered, but opportunities won in TOPMed. Telomeres, they affect many aspects of human health. Longer telomeres are generally associated with more increased cancer risk. Telomere attenuation is generally related to a host of neurodegenerative diseases, but I think most notable is the relationship between shorter telomeres and cardiovascular disease in the literature. But what do we know about telomere genetics? Well, as with that illustration I showed you earlier, telomere genetics is no exception to the lack of inclusion of non-European populations. This is sort of a slide where you see sample size versus the year of publication, color-coded by ancestry. 17 GWASs have been published before 2020, 13 of these in Europeans, 4 in Asian, only a single one in African ancestry, and 1 in Hispanic Latino. But I want to point out that 1 in African ancestry is extremely small in its sample size. This is really important because we do know from extensive prior evidence that telomere differs considerably between human populations and is in fact longer in African ancestry populations, both in comparing African Americans to European Americans, but even looking within continental Africa. So where do we stand on the genetics of telomere length? We know it's a highly heritable trait, 30 to 80%. We know it differs between populations. There have been plenty of GWAS loci. This has actually grown rather rapidly recently with the new UK Biobank initiative. Prior efforts are all using laboratory-based assays, Southern blot, qPCR. What we know is that cumulatively these GWAS signals account for 2 to 3% of trait variants, but as of now there really hasn't been a sequencing-based approach applied to telomere genetics, and so we really know very little about causal variants underlying these GWAS peaks. I mentioned a little bit about the Southern blot and the qPCR, and I just want to give you a snapshot of the technologies and the tools we use to measure telomere length. Without going into much detail, the point here is just to say that there are a variety of tools. The most common use for large-scale epidemiological studies is the qPCR. It's fantastic in that it's high throughput, inexpensive, requires low amounts of DNA, but it has some challenges in terms of it being a relative quantification. It's a PCR-based approach and potential for high variability in the assay. So here comes TOPMed. We've sequenced 140,000 individuals in a real diversity of backgrounds as shown in this pie chart. And all of a sudden now we have the opportunity to address population diversity limitations of prior GWASs because we can leverage this whole genome sequencing data now to actually estimate telomere length bioinformatically from the data and to perform genome-wide GWASs of the WGS-generated telomere length. So really a 2-for-1. We're generating a phenotype using the pre-existing sequence data, and we're really going to be able to help address gaps with respect to the lack of multi-ethnic representation in prior studies. What about estimating telomere length then from the WGS data? This is sort of an area of opportunities sought. And the first question, of course, is can we do this at scale in large resources with sequence data? Because remember, these large consortia level data sets, they are including human genome sequences generated cross-sequencing centers, leveraging legacy DNA, etc. So of course, while we know we can do it bioinformatically, the question is, can we do it accurately, somewhat accurately at least, and can we do this at scale with these large databases? So this was an opportunity we sought in TOPMed, and we applied 2 different telomere calling methods, the method TelSeq and the method CompuTel. And really the primary difference between the 2 is CompuTel costs us a lot more in computational time because it has a realignment step. The first thing we did is to compare these bioinformatic tool estimations of telomere length from whole genome sequencing to FlowFish, which is truly like the gold standard here of telomere length estimation. And in a very small sample set where we don't have major sequencing artifacts, these were 20 samples, freshly collected DNA, all sequenced together at the same sequencing site. We actually had remarkably high correlation in the telomere length from the WGS data, TelSeq on the left-hand side, CompuTel on the right-hand side, and FlowFish data from 2 different cell types. And as you can see, the correlations are really high at about 0.8 and higher. So this looks great, but this is a small data set. No major headaches of sequencing artifacts. Here's a slightly larger data set. This is about 2,500 samples from the Jackson Heart study in TOPMed. And over here, what you're looking at is the correlation in TEL-seq, that's panel A on the left-hand side between CompuTel and TEL-seq. And we had really high correlation in the telomere estimates between the two. The second point is they're both somewhat similarly correlated to southern blot data, which, as I pointed out earlier, is a laboratory assay used to measure telomere length. And on the right-hand side, however, you see that there's a major computational trade-off. So the ultimate decision then within TOPMed was to run TEL-seq on all available samples as the final choice, given this computational cost and given that it performed equally well between CompuTel and TEL-seq compared to southern blot. However, what I'm showing you now is that very same plot layering in colors, and these colors represent sequencing plate. And it's immediately apparent that when you go from a single study with a small sequencing design to a larger study where sequencing was done across multiple plates and legacy DNA, there's a clustering by color. And this clustering by color is a sequencing technical artifact that we discover when we use this sort of large level of data. So this is the difficulty we then encountered in trying to do this in TOPMed. However, we were able to sort of overcome this difficulty because we used a batch correction tool where we are using sort of a correction across the human genome, measuring average sequencing depth every 1,000 base pairs. And we were able to improve that correlation between southern blot and TEL-seq from about a 0.55 to a 0.68, as we're showing you over here. So honestly, we were really, really excited by our ability to overcome some of the sequencing artifacts that we showed you on a prior slide. The second point I think that's really exciting is when you look at these two phenotypic measures of telomere length, one measured from the whole genome sequence data, the other measured in a laboratory assay that has really high quality, the southern blot, they're genetically very highly correlated in a family-based sample from Jackson Heart, and that genetic correlation is 0.8. So that's sort of a second confirmation that these two things are really sort of measuring the very same underlying trait and phenotype that we're interested in acquiring. So how did TOPMed help us? Before I get into this Manhattan plot of then studying this telomere length we calculated on 110,000 individuals, I want to remind you of this pie chart in TOPMed. This is the 109,000 individuals that we're able to study, and put this in the context of these two prior images I showed you before. The left-hand side showing how much of a difference our study in telomere length is compared to just the general catalog of GWAS associations, that little blue pie representing African ancestry. Here, almost a quarter of our data is African ancestry. And the sample size going from this little blip here of 500 individuals now up to 30,000 individuals. If we just think about the African ancestry component, Hispanic, Latino, there's not even, they don't even factor here on this right-hand side in terms of prior genetics. So this is sort of an exciting opportunity we've created, just innovatively thinking outside the box from whole genome sequencing data generated in TOPMed. This is the Manhattan plot, and we have plenty of discoveries. I just want to sort of orient you. This y-axis is split. What you're looking at is p-values that go up to 10 to the power negative 10. The green line is 1 times 10 to the power negative 9. That's sort of our threshold. And what's shown is the loci in red are previously known, the loci in blue are entirely novel. So, you know, this sort of innovative, creative way of leveraging whole genome sequence data to do something really quite novel and measure a brand new phenotype of telomere length. This is this sort of the part of the dynamic human genome, as I referenced, all of a sudden has really added to our biology and genetics understanding of telomere length variability in human populations as a general discovery. These loci actually amount to 36 in total. There are 59 credible sentinel variants in here, and we're able to hone in on sentinel variants, as I'll show you, because we're using whole genome sequence data for this GWAS. 20 of these loci are novel. But of course, what's the value of this WGS? We're zooming in under the hood, if you will, and that's the value to me over using the prior GWASs. This is a series of 10 loci where each little green line represents an independent signal. And so for 10 of these loci, we're able to sort of hone in under the peak and really identify sentinel variants that are independent from each other. So we go from loci now to actual SNPs. And I want to sort of use one illustration, which is TERT. We've thought of TERT here based on the left-hand side in prior GWASs as a single locus. But this opportunity has taught us that there's many SNPs involved in this single locus here. There's 5 independent signals all mapping for TERT. The truth, however, as I started out, is if we're going to think about PRSs, what do these look like? PRSs off of telomere length, what do these look like when you look at them in the context of multi-ethnic representation? So the left-hand side is just showing you what we made in terms of discoveries, putting all of TOPMed together. And then the right-hand side is splitting out the various ancestry, race, ethnicity strata in TOPMed. And you can clearly see, like if you just look at the purple variant, for example, the purple variant is the peak variant in the overall group, but it's not necessarily the peak variant in each individual group. And in fact, if you look at the African Americans, the top right panel, it's not by any means it's the 3rd or 4th strongest association signal. So, this is that idea of saying how important, or reiterating how important it is to not just look at everything together, but tease these things apart. So we used whole genomes to calculate telomere length. Are we finding things that are biologically meaningful? Here's sort of a gene set enrichment using these loci. I've just simply taken it in the top 10 or 20 gene sets. There were 100 that were very strongly significant. And I just underlined the word DNA or telomere, and literally everything in the top 20 is either telomere or DNA. So yes, these genes are definitely highlighting pathways that are telomere biology related. Are these effects big or small? Well, if you look at the effect sizes that we have discovered from this whole genome sequencing approach, we would argue they're quite important. Telomere length seems to decrease at the average rate of about 25 base pairs per year in an adult. In TOPMed, we see effect sizes of about 13 to 1038 base pairs per allele. So, this could cumulatively add up to a pretty significant difference in telomere length based off of these effect sizes directly. What about ancestry differences? So, I showed you just peak SNPs that were different based on which population we looked at. But here's an illustration at the TNF2 locus, where if you look at the top white panel, which is the European ancestry group in TOPMed, you don't even see this association. And so had all of our telomere work been focused on European ancestry populations, we'd never have discovered TNF2, at least not in TOPMed. And this is sort of a really important piece to drive home. Expanding discovery to non-European populations allows you to identify novel signal that's not captured in leveraging only European ancestry populations. But it's even more important than that. We actually have multiple variants where there's true genetic heterogeneity across the ancestry buckets. So, this is not just a feature of power and allele frequencies, but it's actually a feature here, as we're showing you in TOPMed, a matter of the actual effect sizes. Of course, we used bioinformatic tools to compute a phenotype based off of whole genome sequence data. So the question we get asked a lot is, that's really neat, but do you replicate the prior associations? And more importantly, do your novel associations replicate in other data sets that use standard laboratory tools, or are these just some artifacts of sequencing? what I'm showing you here on the left-hand side is comparisons of effect sizes across our 59 sentinel variants to prior publications that use qPCR. And these red dots represent the loci that are novel. These are the loci in our data that haven't previously been found before. And as you can see, they're really highly correlated. So A, they replicate at a p-value threshold for replication, but B, they're also really highly correlated in effect size, which is what you're looking at on this graphic. A second level of saying do they correlate is what you're seeing on the right-hand side. Here we just limited our data to the Jackson Heart study, a single study that I showed you earlier. You've got Southern blot data, you've got TELSEQ data, and if we limit it to the single study and recompute the effect sizes are correlated at an R of 0.92, right? So clearly we're able to measure, leveraging this whole genome sequence data, a quantification of telomere length that appears to work really well. And I want to say actually a LD regression score approach that takes our European ancestry GWAS and compares it to prior published GWASs. So just looking at the summary statistics between two GWASs, one on TELSEQ-derived telomere length and the other on qPCR-derived telomere length, yields a genetic correlation of 0.81. So really, you know, there are so many different levels of validation of this sort of approach we've taken to measure telomere length. But there's so much more, and sadly, I need to start to sort of summarize it. So I'm just going to quickly give you brief snapshots of the so much more without going into too much detail. The first one is that this sort of an approach using whole genome sequencing data has really enabled us to hone in on rare variants and identify, first and foremost, that there are rare variant signals contributing to genes that we've previously implicated from the common variant, and that's what's up here in green. And then what's down in the bottom in red is these genes that we're able to identify that have only rare variant signal and are not discovered by common variant GWAS approaches. Both of these genes actually are really intriguing biological candidates for telomere biology. So that's super exciting. The second so much more piece I want to bring up here is we do have polygenic risk scores that we've created off of the sentinel variants, the 59 sentinel variants, and we've applied this to telomere length PRS to BioVU biobank data. And what we're able to confirm is these previously documented findings that a PRS for telomere length generally is associated with increased risk for cancers. These sort of upward pointing triangles represent increased risk for the outcome. While our associations with coronary artery disease were not significant, I do want to point out that in general, you see this reverse association that I pointed out before, which is all of the triangles that are red are actually flipped and pointed downward. So there's sort of this directional difference between cancer-related risks for telomere length and cardiovascular-related, and these are previously documented and confirmed from the genetic approaches used in this particular sort of opportunity. An additional piece on this polygenic risk score is that when you compute this PRS in the BioVU African Americans and European Americans, we find something really interesting, and that is that the polygenic risk score population distribution in African Americans from BioVU is shifted to the right. So this is, again, really, I think, confirmatory evidence of the phenotypic findings, right? We started out learning that telomere lengths are longer in African ancestry populations. And what we're seeing here is that this longer telomere length in African ancestry populations can indeed be explained in part from the genetic mechanisms that determine telomere length at a population level. And I think this is sort of a really exciting bridging of the gap that we've had thus far in telomere length genetics. There is really, you know, other parts of the dynamic genome that I want to circle and wind down with. And these are, again, opportunities to think about with the massive amount of genome sequencing data we're creating here across all of these consortia level initiatives. And that is to really think about the dynamic parts of the human genome that come from telomere length, as you've just heard about, but also clonal hematopoiesis, and the prevalence of clonal hematopoiesis in driver genes. And yet again, another level would be thinking about mitochondrial DNA, copy number, and heteroplasmy. All these three pieces can actually be called just by having whole genome sequencing data at hand using bioinformatic tools. So this has been done, and there's extensive collaborative effort, again, across various working groups in this particular consortium. And, I just want to hone in on a few additional exciting results to think about for us here. Clonal hematopoiesis was called. We were able to quantify in TOPMed sort of carrier clonal hematopoiesis by driver genes and document this, show that CHIP is, of course, correlated with age, greater CHIP observed at greater age. But work going on to actually study the GWAS of CHIP. So just as we did in the prior illustration where we calculated telomere length and then ran a GWAS of telomere length, both sides of it coming from the whole genome sequence data, here this work was led by Alex Bick and Pradeep Natarajan and many others in TOPMed, where they called CHIP and they ran a GWAS for CHIP. And this is the Manhattan plot from this publication now. And what's exciting is that they landed on two genes that we've known or we've shown as well in the GWAS for telomere length. So two parts of this dynamic human genome component that we're measuring that are coming back to the same genetic loci. So this work has since been extended, looking at the clonal hematopoiesis or CHIP in conjunction with telomere length across these samples. And what we were able to show is that they're sort of here looking at the left-hand side, that CHIP across these driver genes in general is associated with shorter telomere length. You see it here for overall, which is the upper little black circle, but you see it based on the driver genes themselves. If you look, in fact, at the number of CHIP mutated genes, you see sort of this, overall decline in telomere length. So it's a really fascinating sort of bridging and bringing together of two parts of the dynamic human genome using these next generation whole genome sequencing projects and data sets available. And what we've discovered is sort of this bidirectional Mendelian randomization by bringing together these data sets. The first is that Mendelian randomization studies indicate CHIP causes shorter telomeres, which is what you're seeing here. But also, we've noticed that we've been able to identify that the Mendelian randomization studies would support that longer telomere length can, in fact, cause CHIP acquisition. So I want to wind down with a final take-home message and slide, and that is we have created a wealth of opportunities with next-generation sequencing data. We typically tend to try to, at least from a genetics perspective, we're trying to connect genetics here on the left-hand side to clinical phenotypes and endpoints of interest, not just use aging-related phenotypes here as an illustrative purpose, right? It could be any phenotype we're interested in. We're generating all of these massive amounts of omics data, transcriptomics, methylation, lipidomics, proteomics, metabolomics. And we sort of try to bring together genetics and phenotype, incorporating these relevant pieces as a functional component. So sort of upweighting these genetic connectors based on their functional relevance by integrating these additional omics. We're being really creative in this space, maximizing the use of the data using imputation approaches. But I think what we've really identified here in this particular illustration, thinking a little bit more creatively outside the box, is you can actually push the envelope even further to generate whole new phenotypes, right? And here, what we could do is really think about leveraging omics to quantify new phenotypes. We've used the illustration of telomere length. You could think about measures of biological aging, et cetera. But really, I think also to consider and keep in mind jointly leveraging omics from initiatives such as TOPMed to not just expand the use of the data in these innovative ideas, but also to bridge gaps in health disparities by the inclusion of multi-ethnic representation. So I wanted the final slide to truly acknowledge the extensive teamwork involved here. From the telomere work specifically, there's a core team of analysts and investigators, and you know, I just want to say that team science has been amazing and created opportunities that have really pushed the boundaries of our use of the next-gen sequencing data we have in hand in TOPMed.